the floor to Professor Geuna, and then I'll take and start the webinar.
Great.
You just tell the ambassador when he has to start.
Okay, it's fine. Mr. Ambassador, the floor is yours.
Thank you very much. Dear Professor Geuna, dear speakers, dear participants, it is my pleasure to greet you all at the opening of this stimulating webinar, Italy-Japan Approaches to Neural Regeneration. This is another webinar in the series organized by the Embassy of Italy using digital media, but the progressive easing of the travel restriction allows us to be hopeful that we will soon be able to resume events in presence, welcoming Italian scientists in Japan again. The webinar is part of a series organized by the Embassy of Italy in Tokyo through our scientific counselor, Professor Enrico Traversa, under the framework of the Italian Research Day in Japan, and is co-organized with the Neuroscience Institute Cavalieri Ottolenghi of the University of Turin. Let me thank the team of this prestigious Italian university for their collaboration, and in particular for his invaluable help, Professor Alessandro Vercelli, who is also president of the Italian Neuroscience Society. This webinar is also a concrete initiative carried out under the Memorandum of Cooperation in the field of health and medical sciences between the Italian Ministry of Health and the Ministry of Health, Labor and Welfare of Japan. The agreement, signed in 2020, foresees exchanges between the two countries of best practices and know-how in the medical fields. In our views, research and innovation, both at academic and industrial level, are top priorities for developing the relationship between Japan and Italy in the next decades. And we are concentrating our efforts to promote collaborations and joint projects in all areas of mutual interest, particularly in the sectors where our scientific communities share knowledge, competencies, and technology of excellence. In this perspective, neuroscience is definitely a priority sector where Japan and Italy are among world leaders. In a previous event at the Italian Institute of Culture in Tokyo, we highlighted the work of Italian researchers operating in this field in Japan. With this webinar, we wish to continue the exploration of neurosciences, considering neural regeneration. Injuries of the peripheral nervous system often do not spontaneously recover, and stem cell therapies are gaining attention for nerve repair therapies. Our speakers will explore this topic, sharing their knowledge, studies, and observations. I'm looking forward to hearing our outstanding speakers today, and I'm sure that this initiative will greatly contribute to fostering collaborations between Italy and Japan, two countries that strongly believe that advances in the medical field are of utmost importance to accomplish the Sustainable Development Goal number 3, to ensure healthy lives and promote well-being for all at all ages. Please enjoy the webinar. I wish you a very productive discussion and an exciting learning experience. And I'm now pleased to give the floor to Professor Stefano Geuna, Rector of the University of Turin, who is also a neuroscientist. Thank you.
Thank you. Thank you very much, Mr. Ambassador. Thank you. First of all, I would like to thank you, your team, and the organizers of this very interesting and important event. As you already said, Unfortunately, in the last 2 years, we could not move as we are used to, and certainly this is a problem. But science is not stopping, has not stopped. Science continue to go on in the laboratories, even if with more difficulties. And today we look forward really for the next month, the next year, to have the opportunity to get back, to travel and to visit the different laboratories. And in this view, the collaboration between Italy and Japan, especially in the field of neuroscience, have been, and I'm sure will continue to be, very, very strong. As you said, I'm particularly pleased to welcome everyone and give this beginning address of this webinar, because this is also my field of research. So my scientific activity has always focused on neuroscience and especially on neuroregeneration, and therefore I'm particularly pleased that the webinar focuses on this important field. As you mentioned, the nervous system is a very complex system of our body, the most complex, and unfortunately the lesions that can occur either for pathologies or for traumas or for other reasons do not allow a complete recovery, even if our treatment have done very big progresses over the last years. So really we need research, we need innovation, we need new strategies to make the treatment of nerve lesions of the central and peripheral nervous system much more effective. And in this very interesting webinar, the different speakers will tackle this problem from different points of view, addressing different pathological conditions. With, I'm sure we will learn a lot, and the talk that we will listen today would be really very stimulating for all of us to improve and to give new ideas for make our research better and really contribute together to the progress of medicine, and especially in this specific field. So again, thank you very much for organizing this event, and me too, I look forward for a very stimulating webinar, and I'm sure that everyone will really have the possibility to learn a lot from the webinar. Thank you very much, and good webinar to everyone.
Thank you very much to the ambassador and the rector Geuna. I'm Enrico Traversa, the Scientific Council of Italian Embassy in Tokyo. So I will start to introduce the scientific speakers of this event. So we are having 4 distinguished speakers, and we really hope that, as said before, this field needs advancement for bringing new novel therapies to people, and we hope that this can come also through the collaboration between Italy and Japan. So we start now. We will have first 4 scientific talks, and then there will be a panel discussion, which will be moderated by Professor Vercelli. So I now give the floor for the first talk to Dr. Ryoichiro Kageyama, Director of the Riken Center for Brain Science, and he will talk us about functional rejuvenation of aged neural stem cells. Please, Professor Kageyama.
Thank you very much for your kind introduction. So please share my slide. Sorry, I cannot share my slide, unfortunately.
Maria Silvia, we cannot hear you.
Okay, sorry. I gave the authorization, and you should be able to share your presentation.
Yeah, unfortunately,
下になんかコンディビティっていう
It's yes.
3つある
It-
中の一番右が
It's a button near to start video. This is have a first
share.
audio, video, and then sharing.
コンディビティっていうやつですね。
シェアのところはやってるんですけどね、出てこないな。なんで
Condividi.
Okay. It's the third button starting from your left hand side.
I push the third button, but my slide
yeah,
doesn't
but
appear.
it is black, yes, actually, also from me. It is dark.
It seems to be not active and inactive.
Not dark. Yeah, it seems to be inactive also for me.
Still preference. Sorry.
Yeah,
My
now it seems possible from my side.
That it just takes time, might just be slow. So now it seems to me to be active, the sharing
Yeah,
button.
yeah, for me too. いや、コンディビティっていうところ、今白くなってますけど、そこを押されるとできます。
シェアじゃなくてですか？
You told you
Yeah.
you were able to share.
Yes, Marco as well. So
Okay.
it takes now, Professor Kageyama, you should be able to press the share button and
I push share button.
okay, and now are you asked to either share your screen or PowerPoint presentation or something like that?
Just a moment. Okay. Sorry, unfortunately, it doesn't work. So maybe would you,
I think probably we should now start with the second talk, and then
that will
when
be
the problem is
great.
solved, we will be back to Professor Kageyama.
Otherwise,
Yes.
maybe that if you want to send me the slides, I can share myself and I can progress with the slides when Professor talks. Maybe if you, while you are doing the first talk, you can send me the slides and I will share for everybody. Okay.
Okay.
Okay, so I introduce now Professor Annalisa Buffo, who is also belongs to the Neuroscience Institute Cavalieri Ottolenghi of the University of Turin. And she's going to talk us about striatal neuron transplantation for cell replacement in Huntington's disease, prospects and challenges. So please, Annalisa.
So thank you. Can you see my screen?
Yes.
Okay, that's very good. It's a good start already. So yes, I would like also to thank all the organizer for this wonderful opportunity, and of course the colleagues and the attendees that joined this exciting event. It's a very nice occasion to share and discuss the science, and also it's a very important occasion to build bridges, which is something that we really very much need all the time. So today, in the frame of the general theme of neural regeneration, I will present part of the work that we are doing in the lab in the attempt to develop cell replacement strategies and approaches in Huntington disease. And we do so by investigating the capability of human cell derivatives that are produced, that are originated by pluripotent stem cells, namely human embryonic stem cells, that have opened new avenues in the field because of their capability to be expanded in vitro in a dish, and an ability to be guided by the exposure of specific factors and specific conditions to generate human neural progenitors and defined neuronal or glial cell types, depending on the needs of the researchers. So we use these models. We are interested in these models because by means of that, striatal human cells, human neurons can be generated, and we are interested in investigating their therapeutic efficacy in vivo in preclinical rodent models of Huntington disease. And before going to the details of the experiments of the results, I would like to acknowledge the people that are for the most involved in the project itself, which includes my lab in Torino, together with some other colleagues in Torino, you might recognize some, and Elena Cattanio's lab in Milano. Dario Besuso, in particular, in Elena Cattanio's lab. He's an expert in the generation of human striatal cells from human embryonic stem cell lines. And Roberta Schellino and Roberta Parolisi have been working to graft these cells in vivo into animal models of the disease, and have been extensively investigating the capability to integrate and to generate functional benefits. And also this work is performed in the frame of a European consortium, which is supported by Horizon 2020. And this gives us the opportunity to interact with other colleagues that are studying similar cell replacement approaches for other diseases, such as Parkinson's disease or stroke, for instance. So what is Huntington disease and why Huntington disease? Actually, Huntington disease is a severe neurodegenerative disease which is caused by an autosomal dominant mutation in the Huntington genes, and the mutated huntingtin affects very much neurons that are sitting in the corpus striatum, which is this part, very deep in our forebrain. This part of the brain is populated for the most by a type of neuron, which is called the medium-sized spiny neurons that you see here highlighted in green. These neurons are multipolar, and they are characterized by processes that display many protrusion spines, which are synaptic contacts, and that's the reason of their name. There are other neurons that are interneurons as well. I'm introducing this terminology because I will use that during the talk. So mutated huntingtin induces a loss of medium spiny neurons that then drives further degeneration and atrophy in the entire brain, as you can appreciate in this picture of a Huntington diseased human brain. And this leads to important and progressive decline of motor and cognitive function. There is no disease-modifying therapy available at the moment. There is great hope for gene therapy approaches, but they are not ready yet. And anyway, they will not solve the problem of the dysfunctional or dead neurons. And for this, human iPS technology comes to help and allows to develop cell replacement approaches. So why Huntington disease? Well, the goal of, in the question of how therapeutic actions can be achieved by cell replacement in Huntington disease constitutes also a very interesting biological and complex problem that might have or can have general relevance. Well, provided, in fact, that we have the right type of neurons produced in a dish, or right types, because we don't know whether we need only to replace medial-spiny neurons or more neuronal type, and provided that these neurons display long-time survival in vivo and they are safe, meaning that they do not generate tumors, for instance, because of aberrant proliferation, well, we are dealing with the reconstruction of a very complex circuit that is defined by a very specific topography. You can see the circuits I'm talking about in these schematics. I will not go into the detail, but these circuits include both local and long-range loops that are originated from two types of medial-spiny neurons, namely spiny neurons expressing the D1 dopamine receptor, D1 neurons, that are at the source of a pathway, which is called the direct pathway, and other medial-spiny neuron types that are instead expressing the D2 dopamine receptors. These are the source of another pathway, which is called the indirect pathways. And these two pathways, through their interaction, define the balance output of excitation of inhibition of the system. So it is not known to what extent such a complex circuit can indeed be reconstructed by in an adult brain, by the adult circuits, and it is not also known, how much of this reconstruction is needed to reach a therapeutic effect, to reach a therapeutic benefit. So with all this in mind, what Dario did in the lab is to generate a human striatal neurons starting from the H9 human embryonic line according to an ontogenic recapitulating protocol that was formerly established in Elena Cattanio's lab. And if I remember correctly, Marco also contributed to the development of this protocol. So basically, these protocols leads to the generation first of neural progenitor cells through dual SMAD inhibition, and then the cells are exposed to other factors, which are important patterning factors during brain development, such as sonic hedgehog. They are also exposed to inhibition of Wnt signaling, so that progressively progenitors lose cortical traits and acquire features of progenitor cells which are sitting in the niche, which is generating striatal neurons, namely lateral ganglionic eminence progenitors that are at the source of medial-spiny neurons, and also medial ganglionic eminence progenitors that give rise to striatal interneurons. So cells progressively, for instance, acquire, as shown here, LGA traits, and by more or less 50 days in vitro, what we have is a population of young neurons that express bona fide medial-spiny neuron markers such as CTIP2, and for about 50%, and we have 10% of cells that we consider fully mature, as indicated by their expression and co-expression of CTIP2, of GABA. Medial-spiny neurons are inhibitory neurons. They use GABA as a neurotransmitter to communicate with other neurons, and they also express DARP32, which is a phosphatase downstream of the two dopamine receptors. So this protocol is capable to produce medial-spiny neurons progenitors, and within 50 days, a relevant fraction of mature medial-spiny neurons. The rest of the cells, they have the features of committed striatal progenitors. However, cells cannot be grafted at 50 days in vitro because they are already too much advanced in their differentiation and die. So what Dario and the two Roberta established in the lab is to graft them at day 20 at the progenitor stage. So cells were grafted into immunocompromised rats, so to avoid rejection problems, and they were monolaterally grafted in one of the two striata that were formerly lesioned by quinolinic acid. This is a toxin that mimics the features of Huntington disease, meaning that it induces the degeneration of medial-spiny neurons. animals and grafts were monitored over time and analyzed at 2 and 6 months post-grafting extensively in terms of neurochemical phenotyping, analysis of connectivity, and behavioral performance of the rats. We also added a cohort of animals devoted to examine the impact of housing in a rich environment of the grafted animals, meaning that, so a rich environment is a housing condition that includes various forms of sensory motor stimulation and social stimulation as well. Animals live in big cages with other co-specific. They are exposed to colored objects and toys. They can run when they want on wheels. So let's say they enjoy life more and they exercise more. And this condition is known to elicit functional and anatomical plasticity, and therefore we use this experimental paradigm to ask whether the exposure to such environment would promote graft integration and functionality. So what about the results? So human striatal graft could survive very nicely over time up to the end of our experimental time. Over time, the volume of the grafts enlarged together with an increase in the size of the cells, which is a sign of trophism and differentiation. And fortunately, this growth of the volume of the graft was paralleled by a decrease of the density of the cells and by the progressing abrogation of cell proliferation as monitored by the expression of the marker Ki67 and decline of the expression of marker as indicated by nesting. So grafts survive well. They do not display aberrant proliferation. They are safe in a way, and they become interspersed with the cells of the host, which is a sign of integration of the graft. It's here. So what about the phenotype of the grafted cells? Over time, the cells progress along differentiation, and at both 2 and 6 months, we found more or less 50% of cells displaying this bona fide marker, which is a transcription factor, CTIP2, for medial-spiny neuron-committed cells. So when we looked at the fraction of more mature cells, we found that at 6 months, we reached about 10% of cells co-expressing CTIP2 and DARP32. GABA was also overall pretty broadly expressed by the grafted cells, and some of the cells displayed features of striatal interneurons, and the expression of such features increased over time, moving from 2 months to 6 months. So we have grafts that include both medial-spiny neurons proceeding toward differentiation and interneurons. We search also for more, let's say, qualifying markers, especially for medium-spiny neurons, and we explore, for instance, the possibility that we could detect markers for direct and indirect pathway. And at long term, we found neurons, human neurons expressing the D2 receptors and the D1 receptor together with other neuropeptides, which are specific of either the indirect or the direct pathway. So during their maturation, medial-spiny neurons also acquire some more defined features that are, let's say, defining the functionality of the system as well. And by the way, it seems that the medial-spiny neurons of the indirect pathway are predominant over the direct one, which might constitute a therapeutic advantage because indirect pathway medial-spiny neurons are considered to be most affected, at least at early stage, by the disease. And also, I mean, differentiation of such type of neurons in the graft could counteract hyperkinetic features that are associated with the disease. So we explore also other types of organization and marker expression within the graft that could be a sign of the progressive maturation and functionality as a developing striatum. So the striatum, as you can see depicted here in this image of a rat striatum is based of compartments of neurons that cluster together and form islands expressing specific markers as the mu opioid receptor depicted here in green. And these islands that are qualified by the expression of defined markers are interspersed in a sea of other neurons that form the matrix. So the islands are called striosome, sorry, I forgot to mention, and are interspersed into a matrix of neurons that express other markers. So we explored a bit of both striosome and matrix markers as well in the graft, and we found that grafted cells are starting expressing striosome markers, as indicated here by the mu opioid receptors. So they are progressing also at the level of this compartmental organization. And the appearance of features of striosome makes sense because medial-spiny neurons belonging to the striosome are considered to be known to be the first one to be generated during development. Then we wanted to explore and understand a bit better about the let's say the morphology of the grafted cells. Our focus is always on medial-spiny neurons. We took advantage of the possibility to obtain sparse labeling by expression of the reporter M. Cherry, which was obtained by viral transduction. And we perform morphometric analysis on M. Cherry expressed grafted human cells. As a result of this morphometric analysis, we found that about 1/3 of all the analyzed human neurons, they display, let's say, features corresponding to authentic human medial-spiny neurons as quantified and analyzed formerly by Gravelan et al. Especially among this population, there was one that comprises about 13% of all the cells that display spine density corresponding to fully mature human medial-spiny neurons. So in other words, this extensive analysis clarified that a long-term human medial-spiny, grafted human medial-spiny neurons display authentic features including both neurochemical and morphological traits corresponding to human medial-spiny neurons. Then we looked at the connectivity. We traced the innervation fibers exiting from the grafts by analysis of human-specific markers such as STEM 120 and the human NCAM, and we found that already at 2 months, neurites from the graft reached relevant striatal targets, depicted here by the asterisks, such as the globus pallidus, substantia nigra, subthalamic nucleus. Fibers were still there at 6 months, with no increase in the density of innervation and no gross aberrant connectivity. So from the point of view of the innervation, we can say that that was pretty efficient and occurred pretty early, and pathfinding seems to be working properly in the context of an adult human brain. Then we explored the input. So we asked whether grafted neurons received afferents. And we did so in collaboration with Malin Parmar. We adopted an approach based on monosynaptic tracing, which exploits 2 different viral systems. I will not go into the detail, but so grafted cells that are potential target of innervation can be distinguished by the expression of a nuclear GFP and by the expression of a fluorescent red reporter M. Cherry in the cell body. At the same time, with this system, we can identify the first-order afferents that contact the grafted cells that we call traced neurons that are labeled by M. Cherry expression transsynaptically. So this kind of approach and analysis allows us to understand that grafted cells receive first-order inputs from striatal areas, including both other human cells and host surviving striatal cells at short time points, and this was further confirmed at 6 months. However, at short time points, we were not able to detect first-order afferents from extrastriatal sites that instead we found at 6 months. And extrastriatal first-order neurons contacting striatal cells at 6 months were found in the prefrontal cortex, motor cortex, thalamus, amygdala, hypothalamus, at sites that are known to be part of the striatal circuits physiologically. So this is also a good evidence of the capability of the graft to become wired into the host circuit. We also explored, in collaboration with Gerardo Biella at the University of Pavia, the functional activity at the cell level of the grafted cells. So already at 2 months, these cells, a fraction of these cells can produce action potential and display voltage-gated sodium and potassium currents. Over time, Gerardo was also able to detect spontaneous excitatory postsynaptic potential, indicating the functionality of the grafted cells. So I mentioned that we dedicated a cohort of animals to the exploration of the effect of enriched environment. So I will just mention, the changes that we found induced by the exposure to such housing condition in comparison to exposure to standard environmental housing. So what we found is a specific effect of environmental enrichment in the promotion of the maturation of graft mediospinal neurons, where we have a doubling in the proportion of mature cells, evidenced by CTP and DARP2 expression. We have also an increase in GABA expression, no impact on the expression of interneuron markers. Also, what we found is that exposure to enriched environment increased the proportion of cells displaying mediospinal neuron features by morphometric analysis, and especially double the fraction of cells with high spine density that correspond to the fully mature adult human mediospinal neurons. So enriched environment promoted the maturation of graft mediospinal neurons, also in morphofunctional terms. It also increased the level of connectivity, the number of input neurons, as you can see and appreciate by the almost doubling of extrastriatal first-order inputs, and by the significant increase in the connectivity index, which is the ratio between the input cells and the grafted cells from the host. And we also observe an increase in the number of TH-positive fibers that are dopaminergic fibers projecting to the striatum from the substantia nigra. So we found important effects of the enriched environment. We also looked at behavior in terms of motor behavior for the most. Upon quinolinic acid monolateral lesion, what happens is that the contralateral forelimb displays akinesia, and we tested the level of this akinesia in a number of sensory motor tests. Some are illustrated here. We found that the graft improved the akinesia of the contralateral affected forelimb. Already at early time points, with no further improvements at later time points or in the enriched engraftment condition. At these early time points, at 2 months, we were not able, though, to detect improvements in more complex motor tasks, such as those that the animals perform on the rotarod where they have to run on a rotating road. However, at longer time points, while the control sham animals that were only affected by the lesion displayed a progressive deterioration of the performance, the presence of the graft, as illustrated here in light green, counteracted such deterioration and ameliorated the performance of the animals. This performance was further increased by the exposure to enriched environment, see the green line here. However, a rich environment per se, in the absence of the graft, also exerted an improvement in the motor ability of the grass, suggesting action which are independent of the action of the graft itself. So let's come to the summary. So overall, this data we think show very nicely the therapeutic value of human striatal graft produced with the protocol which has described. We are dealing with graft which are composite graft that include both mediospinal neuron and interneurons. The identity of mediospinal neurons we think has been qualified by our results in comparison to real and authentic human mediospinal neurons. Also, human striatal grafts get wired into the osseous circuits. They display a good degree of circuit reconstruction that is associated with improvement of motor performances. A rich environment is interesting to notice that fosters the morphofunctional maturation of mediospinal neurons specifically and improves graft integration. There is more to be done, of course, in the cast for the most therapeutic graft cells. For instance, we have to deal with the problem of this very slow and asynchronous differentiation pace of the graft in vivo. We would like to understand what is the best graft composition to give rise to the best therapeutic effect. We need to understand the detail of the circuit reconstruction. We don't know to which extent the direct and the indirect pathway are reconstructed, and we don't know what is the role of the activity of the graft in the motor improvement that we found. Well, our data show that a rich environment can be a nice complement to foster graft differentiation and integration. It also suggests that the exploration of the mechanism that underlie this effect can be valuable to promote, to accelerate, to design approach to accelerate the graft differentiation. I'd just like to end by giving credits again to all the people that made it possible: Roberta Schellino, Roberta Palolisi, Gerardo Biella, Dario Besusso, and Malin Parman as collaborators. And thank you for your kind attention, and to our funder for making all this work possible.
Thank you very much, Annalisa, for a very interesting presentation with a nice example of promising, still promising only, but promising example of using stem cells for neuroregeneration. So before passing the word again to to Kageyama Sensei, I would like just to mention that participants can use the function of question and answer for making questions to the speakers that will be dealt in the panel discussion at the end of the presentation. So now I give again the floor to Kageyama Sensei, hoping that everything is fine now.
Let me try. Okay. Just a moment. So. Just a moment. Sorry.
Kageyama Sensei, do you think that we should move to the next talk?
Sorry, again, it doesn't work. Sorry for my trouble. Yeah, please switch the order.
Okay, so let's go to the next talk and then try to solve the technical issues. So now I will give the floor to Professor Tadashi Isa from the Institute for Advanced Science of Human Biology, the WPI Ashby in Kyoto University. He will talk us about the brain mechanisms for recovery after spinal cord injury.
See my slide?
Yes, we can see your slide.
Yeah, and can you hear my voice? Yes.
Yes, we can hear your voice.
Okay. Okay. Sorry. Okay, thank you very much for the invitation, and it is a really great honor. So I'm Tadashi Isa from the Kyoto University, and today I'll talk about the brain mechanism for the recovery after spinal cord injury. So of course, stem cells, today I'll talk about the neural regeneration more like a top-down kind of approach. And so for the, functional recovery, the implanted cells, stem cells should be incorporated into the neural circuit for the proper operation. So I'll talk about so for that purpose, some kind of neurorehabilitation therapy or neuromodulation therapy may help the further recovery. So today I'll talk about that functional viewpoint. So I've been working on the neural mechanism of controlling the dexterous hand movements, like independent finger movements, which developed in higher primates. And so it is generally considered that the corticospinal tract, which originates from the motor cortex and descends through the most ventral part of the brainstem, and the majority of them crosses the midline at the most caudal part of the brainstem, and descends through the lateral nucleus of the spinal cord and connected directly to the motor neuron. So this kind of corticomotor pathway is the basis of this dexterous hand movement. But actually, this is very oversimplified scheme. And it is known that only a few percentage of the corticohural fibers are really directly connected to the motor neurons. And there are also many other parallelly indirect pathways. So this is a schematic diagram of the brain and brainstem and the spinal cord. And so in addition to this direct connection from the motor cortex to the motor neuron, there are also brainstem relays mediated by the red nucleus or reticular formation. And as for the function of this brainstem relay pathway, so there are some studies which transected the brainstem pyramid, in which the corticospinal fibers are transected, but these brainstem relays remain intact. But in this case, like in the Lawrence Kuiper's paper in 60s, the precision grip movements are permanently impaired. So these results suggested that these brainstem relays cannot compensate for the precision. But in addition to the brainstem relays, we have demonstrated that there are also spinal relays mediated by the propriospinal neurons or segmental interneurons. And in the case of these propriospinal neurons, the axons descend more ventral part of the spinal cord compared with the corticospinal fibers. So if we make this kind of region of the lateral nucleus, you can region the direct pathway, but leaving this indirect pathway intact. Then what happens to the monkey? And actually, so this monkeys are trained to perform this precision grip movements, and one day after the region, the monkey can still reach, but their finger movements are impaired. If we continue to train the monkey every day, then in 1 week, still the index fingers are paralyzed. But in 2 weeks, they start to recover, but it still looks clumsy. But in 3 weeks, they become much better. And in 1 month, they look almost normal. So we are using this monkey model and studying the neural mechanism of recovery. So first, I said that this propriospinal neurons, which mediate the cortical command to the motor neuron, may compensate for the lost function. But to demonstrate that, we use a double viral vector technique to selectively block this neuron. So we injected a retrograde viral vector into the motor nuclei, but in this case, the vectors are transported to the segmental interneurons, the propriospinal neurons, also the corticospinal neuron. But if we inject the second vector at the location of the cell bodies of propriospinal neurons, only these groups of the neurons are doubly infected. And upon the administration of the doxycycline, the two genes are interacting each other and synthesize the tetanus neurotoxin, which is transported to the nerve terminal, cleaves the BAMP2, and stops the synaptic transmission. So by using this technique, we could selectively block this propriospinal pathway, and then found that the monkey's precision grip movements are impaired. Like this, the monkeys cannot extend their thumb. But since this major corticospinal fibers are remaining, the impairment of the failure ratio is transient, and movement time is just transiently prolonged. But if we combine this blockade of the propriospinal neuron with the spinal cord injury, then even after more than 4 months after the injury, the recovery is still partial. So the recovery was impaired. That means the propriospinal neurons are involved in the recovery after the corticospinal tract injury. But in addition to this spinal mechanism, we found other more higher level of neural systems involved. So to study that, we made a positron emission tomography imaging of the brain activity during the recovery, and found that during the early stage of recovery, around 1 month after the lesion, in addition to the contralateral motor cortex, the ipsilateral motor cortex hand area is also activated, but which disappears in the long run after more than 3-4 months. Here, the originally activated primary motor cortex is deactivated again. But to demonstrate whether this ipsilateral activity is really involved in the recovery, we injected a GABA receptor agonist Mushimo to this area, and what happens to the monkey's movement. So about 1 month after the injury, monkey recovered to this level. And then we injected the Mushimo into the ipsilateral motor cortex. Then, very interestingly, it impaired the movements. So it demonstrates that ipsilateral motor cortex is involved in the recovery at this stage. And also, in more higher level, we also found in that PET scanning study, we found that the nucleus accumbens activity is also enhanced. So this was particularly interesting because nucleus accumbens is generally considered to be involved in the control of regulation motivation. And everyone knows that motivation is very important for the rehabilitation, but no one knows the mechanisms. So we try to explore the mechanisms, and we also injected Mushimo into this nucleus accumbens to demonstrate its causal contribution. So we injected Mushimo into the nucleus accumbens before the spinal cord injury, like here. The monkey is making the pressure grip nicely, and in this way. And then we injected Mushimo into the nucleus accumbens. But actually, virtually no effect. So the nucleus accumbens may not be so important in the motor control at the normal condition. But in the case of recovery after spinal cord injury, the 16 days after the spinal cord injury, the monkey is starting to recover the pressure grip in this way. Then we injected Mushimo into the nucleus accumbens. Then we found it had a very strong effect of impairing the recovery in the movement. Actually, so this happened only in the left hand, which showed the recovery. But the right hand, the intact hand, was completely normal. So in this way, this is a recovery curve of the success rate of pressure grip in this monkey after this corticospinal tract lesion. So if we inject the inactivate the nucleus accumbens before the injury, no effect. But during the recovery course, it had a profound effect in impairing the recovery movement. But sometime after the recovery, then the inactivation doesn't happen like that. But anyhow, the nucleus accumbens has a critical role during the recovery stage. So in this way, so for the recovery after a partial spinal cord injury, many brain regions like ipsilateral motor cortices or nucleus accumbens, which is not usually used for the normal control, starts to be involved in the function. But anyhow, here I showed about the result of the partial spinal cord injury, in which we selectively transected the corticospinal fluid. So in this case, the propriospinal system, the parallel system helped the recovery. But this is a rather artificial type of the lesion. So in more recent study, we are also studying more larger lesion, like subhemisection injury model. So in the previous studies, so it was known that this lesion model is very hard to recover. So even after half a year, so the monkey's recovery is very, very poor. But what happens in this case? So actually, we implanted the electrocorticography electrodes in the bilateral premotor, motor, and somatosensory motor cortex, and made this very relatively large lesion and studied the monkey's recovery. But actually, we found these monkeys recovered pretty well. So we defined the movements in a precision grip, so picking up the small foot from between the slits, so without any interference. This we define the precision grip, and but without but these monkeys showed this movement never recovered after a long time. But if we remove this slit, they can grasp in a more coarse grip. So we call this grip gross power grip. And also, if we plot use this slit, then the sum of the movements are guided by the slit. So this we call the slit guided pinch. So precision grip never recovered. This power grip recovered in 1 month, and the slit guided pinch was recovered in 2 months, which was very amazingly fast. And then what happened to the monkey's brain? So we applied the electrical stimulation through these electrodes, the 3 milliampere, 3 pulses at 20 hertz, and examine the twitch movements in the hand, in the arm and hand. So this blue color indicates the strength of twitching. And before the injury, the twitch from this premotor region, the twitch could be induced in the back and hind limb and shoulder. They have to be distal part, more proximal part of the muscle. But after the injury, once it's impaired, but actually sometime in parallel to the recovery of the hand movements, it showed induce a twitch very extensively from the distal region to the proximal region. This is just from the one electrode. And from the other electrode from the contraregional cortex, also, as you see, in the premotor, motor, and somatosensory cortex, the stimulation induced the twitching movements, very global. So this is from the contraregional side, and also from the ipsiregional side, ipsilateral side of the spinal cord injury. So it also induced very extensive twitching. So it seems that very widely, globally, the cortical circuit seems to be disinhibited. And so this is the number of the channel of this electrode, which could induce twitching by stimulating. So first, I show you about the effect of contraregional motor cortex and affected hand. So actually, the number of the twitches from the distal muscle to the proximal muscle, or the number of the channel which induce the twitches was increased. So this is from the contra to affected hand. And this is from the ipsilateral cortex to the ipsilateral region. And also, before the injury, no channel induced, but during the recovery, many channels started to induce a twitching. So this is for the affected hand. And also for the non-affected intact hand, also the number of the channel increased. from the, this ipsilateral cortex to the other hand, and also from the contralateral to the contralateral. It doesn't seem to be related at all, but also it seems to be increased. So regardless of the target muscle, the whole brain seems to be rather disinhibited. And to study the, I call this global disinhibition during the recovery. And to study the mechanism, we also use this conditioning test. So to study the interaction between the bilateral premotor motor cortex, the neurologist uses this kind of technique. So first we stimulate the contralateral motor cortex and induce some twitches into the muscle response in blue in this way. And so the ipsilateral stimulation is given sometime, some millisecond before the twitches, and whether it is how the preceding stimulus affects this twitch. And actually, before the injury, this effect, the preceding effect is usually inhibition. The muscle twitch is, suppressed. So that means, the effect is inhibitory. So we call this interhemispheric inhibition. But during the recovery, actually, this interhemispheric inhibition is switched to the interhemispheric facilitation. Also, this shows another sign of global disinhibition. So how should we think about the neural mechanisms? So it is considered that this bilateral premotor motor cortex is interacting, have the both excitatory and inhibitory connection. But in the normal status, the inhibition predominates. So EI balance, excitatory and inhibitory balance, is predominated by the inhibition. But somehow, the activity of this inhibitory neuron might be lower by some kind of factor. Then the excitation dominates. So that will be the reason why the cortical circuit is very globally disinhibited and starts to, elicit the muscle twitches from everywhere. But you may wonder why this happens, because the downstream pathway is transected. So you may wonder why this happens. So to study what is happening in the downstream of this corticospinal tract, we study the trajectory of this corticospinal tract by injecting the viral vector-based neural tracer, green on the affected contraregional side and red in the intact side. Then, as for the ipsiregional motor cortex, which is the intact side, then the corticohumeral fibers that crosses the midline in the pyramidal decussation and descends through the contralateral side. So this is very close to a very normal pattern of the corticospinal tract. But in the case of affected side, so the axons once cross the midline, but they are transected. But surprisingly, you can find many fibers uncrossed or seems to be, switching their route and descend through the contralateral side and descend to the lower cervical segment. Yeah, there is a very predominant difference in the axonal trajectory after this injury. Then we made more quantitative analysis about the number of the axon fibers in each segment. And in the case of the, the unaffected side, more than 90% of the corticospinal fibers are crossing the midline and descend. And also, but in the case of the affected side, more than around 20 to 40% of the fibers are descending in the, ipsiregional side. And the majority of them actually crosses the midline and terminates on this side. So it seems that there is some massive rerouting of the corticospinal fibers, and they cross the midline again, causal to the injury. So this is a camera lucida drawing of the corticospinal fibers at the pyramidal decussation, and also at the C6 segments, which is caudal to the region. So many of the corticospinal fibers descending in the lateral funiculus crosses the midline and then terminates in the spinal cord, including the ventral horn of the neurons. Yeah, so this was really amazing. And actually, so we counted the number of the axons or butons in the each layer of the spinal cord. And for this normal side, they usually terminate on the layer 6, 7 or layer 9 of the spinal cord. But in the case of the corticospinal fibers originating from the affected side, so actually majority of them actually even though they are descending the same part of the spinal cord, they cross the midline and goes to the other side and terminating the lamina 6, 7 and lamina 9, which might have been their original target. Then, yeah, this was really amazing massive plasticity. But then why was the recovery was so fast and such a massive rerouting of the corticospinal tract happened compared to the preceding study? Actually, we can think about the two issues. So one of the differences is that in this study, the monkeys received very intensive training starting a few days after the injury. And also the monkeys received intensive electrical stimulation, 3 milliampere 3 shocks for 100 times for each electrode for all the 18 to 36 channel of the sensory motor cortices to the bilateral premotor, primary motor, and sensory cortex everywhere. So maybe, so this is an intact normal control of the movements. Mainly it is the hand movements are controlled by the corticospinal tract and partly through the propriospinal neurons, indirect pathway. And in the case of partial lesion, this indirect pathway may take over the function. But in the large lesion like this, but usually the recovery is very poor. But through intensive training and electrical stimulation, we could succeed in inducing the massive plasticity in the adult primate brain. So the global distribution to recruit a novel neural system for the recovery. Okay, thank you very much for your attention. And all these are the collaborators of my talk. Okay, thank you very much. I stop here.
Thank you very much, Isa-sensei, for the very interesting talk. Very careful approaches in trying to understand the mechanism, which are very difficult for the brain, for understanding. Kageyama-sensei, what is the situation now? So I think we can continue with the talk of Marco Onorati for the moment, since Kageyama-sensei is not answering. So I'll give you the floor to Marco Onorati from the Department of Biology of the University. Kageyama-sensei, are you ready or not?
I'm trying to share my slide, but I'm not sure. Can you see my slide? Not yet.
No. So I continue with Marco, and then we try to solve the problem. Oh, okay. Maybe yes. Now we have here the sharing. We can see. So we took the success of the moment for giving you the floor. Thank you very much.
Thank you very much, and sorry for my big trouble. But you can see my slide, or it's a.
Yes, we can see your slide.
It's not a slideshow. Let me try it. All right. すいません. Sorry, do you see my slide?
We see your screen at the moment, which is good.
Now we see your screen, not the slides.
Screen.
But if you click on the PowerPoint icon,
I chose PowerPoint, but it suddenly disappears.
Yeah, just click again on the icon.
moment. Actually, everything disappears. Oh.
Try to open again your slides, your PowerPoint.
すみません。ちょっとまたやっていただけますか。ちょっとなんか消えてしまったんですけど。どこ行ったの？ファイルが一瞬選んだんですけど、消えてしまったんですよ。どこへ消えたんですかね。何も出てこないんですよ。ちょっとなんか読んでいいですか。You are sharing.
We can see your screen. So
Screen?
you're sharing the screen, but we are not seeing your presentation.
Oh, what happened?
So probably if you open again your PowerPoint, we should see.
Really? Okay. Cute. Okay, this. Sure.
Yes, now we can see.
Can you see the slide?
Yes, we can see the slides. Yes, now it's fine. It's okay.
Okay, okay, great. Okay, sorry for my trouble. And I'd like to thank the organizers for giving me this opportunity to talk about our work today. So today I talk about utilizing endogenous neural stem cells for future therapy. So I have two topics. In the first part, I'll talk about characterization of adult neural stem cells. And the second part, I'll talk about functional rejuvenation of aged neural stem cells. So we are entering the age society, and people with dementia or Alzheimer's disease are increasing in number significantly. But there's no effective treatment for dementia, unfortunately. So this is a big issue, big problem in many countries like Japan and Italy. But good news is that there are some neural stem cells in the adult brain. So we might be able to use these neural stem cells for regeneration of diseased or damaged brain. That's our future goal. But adult neural stem cells are totally different from embryonic neural stem cells. Embryonic neural stem cells are very active, and they are highly proliferative, and they are multipotent, so they can make the whole brain from scratch. By contrast, adult neural stem cells are mostly quiescent or dormant. They have only limited proliferation ability. They have limited neurogenic potential. So adult neural stem cells cannot regenerate diseased or damaged brain. But still, these neural stem cells can make some neurons and play an important role in brain functions. So here, this shows the mouse brain, and in the adult brain, these two regions contain neural stem cells. One is subventricular zone of the lateral ventricles here, and the other is subgranular zone of the hippocampal dental gyrus here. And these regions have neural stem cells, and these neural stem cells produce some neurons, and which play an important role in brain functions. And here to what extent neurogenesis occurs in the adult brain. So to answer this question, we generated nesting CRE-ERT2 mice. So here, CRE-ERT2 is specifically expressed by neural stem cells under the control of nesting promoter and enhancer. And after tamoxifen treatment, this CRE becomes active, entering nuclei, and deletes this stop cassette, allowing the ROSA26 promoter to express the green fluorescent marker. And so after tamoxifen treatment, here all neural stem cells are labeled with a green fluorescent protein. And also neurons born from these neural stem cells are labeled with the same green fluorescent protein. So here we examined the neurogenesis in the adult brain. And before tamoxifen, after tamoxifen treatment, there's no labeled cells. But after we treated tamoxifen at 2 months of age, young adult stage, and 10 days after tamoxifen treatment, we found the green fluorescent labeled cells appearing here. This is a hippocampal dental gyrus. And in this region, neural stem cells are present along this line. So these data show that neural stem cells are efficiently labeled with green fluorescent protein. And 1 month, 3 months, and 6 months later, we found yellow cells appearing in this region. So neurons born from these labeled neural stem cells migrate into the red zone and become yellow. So we quantified yellow cells. So those are newborn neurons in the adult brain. And we found about 10% of the neurons in this region are born in the adult stage. So suggesting that relatively many neurons are born in this region. And we next examined the functions of these neurons. So to answer this question, we ablated all newly born neurons by using this strategy. And again, we use a nesting promoter enhancer-driven QERT2. And the target genes here, neuron-specific promoter, stop cassette, and diphtheria toxin. And after tamoxifen treatment, the neural stem cells activated Cre deletes this stop cassette. And now diphtheria toxin is ready for expression. But as long as these cells remain as neural stem cells, this promoter is inactive. So neural stem cells are intact. But once these neural stem cells start neuronal differentiation, then this promoter is active and expressing diphtheria toxin. And the diphtheria toxin instantly kills the cells. So in these mice, after tamoxifen treatment, all newly born neurons were specifically killed and ablated. So by using this strategy, we blocked neurogenesis completely. So here we examine the histology, and the DCX is newborn neurons in the adult brain. And without tamoxifen treatment in the control, here DCX positive young neurons are present like this, very abundant. But when tamoxifen was administered, so neurogenesis was blocked, then there are very few DCX positive young neurons here. And we quantify the total neuronal number, and the open bar shows the negative control, and we found the neuronal number is increasing with aging, like this. But here, closed bar show the mutant, so whose neurogenesis is completely blocked. Then the neuronal number was not changed. So neurogenesis does not occur anymore in these mutant mice. And we found here the histology seems very similar, but we found fewer neurons in the mutant than the control. And we next tested Barnes maze test for spatial memory. And we used this kind of open round table, which has 12 holes at the periphery. And one hole has a small bag beneath, but the other 11 holes are just holes, so they are very dangerous. Mice may fall onto the floor. And if you put a mouse on this table, they don't like this kind of open space, so they try to hide these small spaces. And they try each hole, and they finally find a hole with a small bag underneath, and they enter it because they don't like this kind of open space. And the position of the target hole with a small bag is fixed. Then after everyday training, these mice gradually remember the position of the target hole. As shown here, control mice showed fewer and fewer errors day by day, as shown here. By contrast, the mutant mice whose neurogenesis is completely blocked, they showed more errors than the control, indicating that the acquisition of spatial memory is impaired by blockade of neurogenesis. But still, they gradually showed fewer errors after everyday training. And we found that after 1 week everyday training, both control and mutant mice remember the position of the target hole. Then we tested 1 week later without any further training. Then we found control mice still remember the position of the target hole, but mutant mice didn't remember the target hole anymore. So these data show that the retention of the spatial memory is also impaired by blockade of neurogenesis. So these data indicate that neurogenesis is very important for spatial memory. And we also found neurogenesis is very important for other brain functions, like male sexual behaviors and female nursing behaviors, and so on. I show one example here. It's a Barnes-Maze test. So this is a control mouse. His target hole is here. Well, he made a few mistakes, but he remembers this target hole very well. And this is a mutant mice. His target hole is here. You see, he doesn't remember his target. He totally forgot. He's almost giving up. Yeah, now he enters it. So these data show that neurogenesis is definitely required for spatial memory. Okay, so next I'd like to talk about functional rejuvenation. So continuous neurogenesis is very important for many brain functions, including spatial memory and other behaviors. But neurogenesis declines during aging, as shown here. This is, again, shows hippocampal dentate gyrus. Here, neural stem cells are present along this line. And at 6 months of age, which is relatively young, corresponding to 30 years old in humans, neural stem cells are relatively activated and forming many DC positive young neurons. So these neurons are very important for brain functions. But formation of DC positive young neurons decreases with aging, as shown here, 9 months, 12 months, and 18 months of age. At this stage, virtually no DC positive young neurons. At this stage, there are still many neural stem cells here, but these neural stem cells are rarely activated at this stage. So we try to activate these dormant neural stem cells to enhance neurogenesis. And to this end, we first compare the transcriptome between embryonic neural stem cells and adult and aged neural stem cells. And we identified embryo high genes and adult or aged high genes. And we screened for genes that can activate quiescent neural stem cell cultures by overexpressing embryo high genes and knocking down adult or aged high genes to identify, to know whether any gene set can efficiently activate aged neural stem cells. And finally, we found this combination, overexpressing the embryo high gene PLGLU2 and knocking down the aged high gene DRCON-A. This combination, we call it IPAD for short, inducing PLGLU2 and anti-DRCON-A. IPAD treatment can efficiently activate aged neural stem cells, as shown here. This is 19 months of age, but IPAD treatment can efficiently activate these neural stem cells, and these neural stem cells proliferate and make DCX positive young neurons. And here we quantified how many cells are produced from single neural stem cells. And this is a control, 9-month-old control neural stem cells. Here, about 70% of neural stem cells did not divide during the 3-week tracing period. And 30% of neural stem cells divide only once, so forming 2-cell cluster. So proliferation ability is very low. This is 19-month-old aged neural stem cells. More than 90% of neural stem cells did not divide at all during the 3-week tracing period. By contrast, IPAD-treated neural stem cells, they proliferated very well, forming clusters or consisting of many cells, like 10, 15, or 20 neurons. So these data indicate that IPAD treatment can efficiently activate the neurogenic and proliferation ability of aged neural stem cells. And we also quantified the neurogenesis. And here we injected either control virus or IPAD virus at 18 months of age and examined brain sections at later stages. And here, when the control virus was injected, virtually no activated neural stem cells appeared at later stages. By contrast, when iPad virus was injected, we found many activated neural stem cells. So iPad can efficiently activate neural stem cells. And we also quantified newly born neurons here. So when control virus was injected, there are very few newborn neurons. But when iPad virus was injected, we found many newborn neurons. So indicating that iPad can enhance neurogenesis in the aged brain efficiently. So we also examined aged markers here. Here, this is a 1-month-old young neural stem cells, and this is aged neural stem cells. And we examined the beta-gal activity. Beta-gal activity is very low in young neural stem cells, but very high in aged neural stem cells. But these aged neural stem cells were treated with iPad virus, then beta-gal activity was decreased significantly. We also examined other aged markers like P16 and P19. These are very low in young neural stem cells, but very high in aged neural stem cells. But when iPad virus was infected, these aged gene expression was significantly decreased. So all these data indicates that iPad treatment can reduce the aged gene expression here. So suggesting that iPad treatment can rejuvenate aged neural stem cells. So here we performed a transcriptome analysis. This is embryonic day 13 neural stem cells, and this showed 1-month-old young active neural stem cells, 1-month-old quiescent young neural stem cells, 9-month-old and 20-month-old aged neural stem cell, showing transcriptome changes with aging like this. But when these aged neural stem cells were infected with iPad lentivirus, then they became very similar to 1-month-old young neural stem cells. So indicating that iPad treatment can efficiently rejuvenate aged neural stem cells. So we performed a behavior test, and this is a Barnes-Maze test. And black line showed the control aged mice, and the blue line showed the iPad-treated aged mice. And we measured how long they walked before they get to the target hole, and how long it took to get to the target hole, or how many errors they made before they get to the target hole. And all these scores showed indicates that iPad-treated aged mice showed better scores than control-aged mice, indicating that iPad treatment can improve the spatial memory of aged mice. And here we tested a novel object recognition test. And we put a mouse in a small room containing 2 identical objects, A. And after 10 minutes, each mice was returned to his home cage. And 24 hours later, each mouse was put in this room containing object A and novel object B. And the mice usually are more interested in novel object, so they spend longer time near object B. But aged mice didn't show much difference between these 2 objects. But when these aged mice were treated with iPad, then they showed more interest in a novel object, so spending longer time near object B. So these data indicates that iPad treatment can improve the cognitive functions of aged mice. And we also found the neurogenesis is severely impaired in Alzheimer's disease model mice, but iPad treatment can activate neurogenesis in Alzheimer's disease model mice. And also iPad can improve their cognitive functions. So all these data indicates that iPad treatment is very promising for the future therapy of dementia or Alzheimer's disease. So this is a summary. Neural stem cells are present in the adult brain and occasionally produce new neurons. And neurogenesis occurs in the adult brain. And neurogenesis in the adult brain is very important for learning and memory. And neurogenesis declines with aging and Alzheimer's disease. But iPad can rejuvenate aged neural stem cells and enhance neurogenesis. And enhanced neurogenesis ameliorates cognitive functions of aged mice and Alzheimer's disease model mice. So iPad treatment seems to be very promising for the future therapy. Okay, lastly, I'd like to thank my colleagues and we have recently moved from Kyoto University to RIKEN CBS. So we are now looking forward to this new research in this institute. Okay, so that's it. Thank you very much for your kind attention.
Thank you very much, Kageyama Sensei, for a very interesting talk, very fascinating. So hopefully that can be really, if we can rejuvenate, that would be a real promise. So now let's move to the last talk since we are quite late. Now I give the floor to Marco Onorati from the Department of Biology of the University of Pisa. And his talk is entitled Neural Stem Cell Transplantation and Stretch Growth, a combinatorial approach to address spinal cord lesions. Marco, the floor is yours.
Yes, thanks. So good morning or afternoon, everybody. Thanks a lot to all the organizers for giving me this great opportunity to be here. And now I'm trying to share the screen. Can you see, please, the slides?
Yes.
Shared. Okay, good. So I will try also to put the laser pointer. Okay, so today I will talk about the combinatorial approach that we are considering to address spinal cord lesions that consist in putting together the neural stem cell transplantation together with nanomedicine tools that are basically promoting the growth of axons through mechanical stretch. Movement is the way we communicate to the world, and the motor controlled execution is one of the most important activities we do that allows us basically to exert our actions on our surroundings. Unfortunately, a number of pathological conditions, as already anticipated by my colleagues, may affect this ability. And in particular, I will talk about the spinal cord lesions. Just to summarize briefly, spinal cord lesions can be both traumatic or not traumatic, and the severity depends on the level, on the segment of the spinal cord that is being injured. So lower lesions can affect the, for example, bowel and bladder function, but going up to the lumbar level, so patient may experience paraplegia, or even if you move toward the cervical portion, unfortunately, patients are experiencing very, very devastating effects like tetraplegia, and sometimes they are not even able to breathe autonomously. Usually also the patients are young, statistically also males are more affected, and so they have an entire lifespan that is condemned to paraplegia sometimes or tetraplegia. There are no resolutive cure at the moment because, you know, the compressive surgery or other treatment that are not able to have a very, very long-term beneficial effect. So what is the rationale of cell transplantation? So traumatic spinal cord damage results in the cell loss at the injury level, as well as a disconnection of surviving neurons with an irreversible interruption of the information flow to and from the brain. The implantation of neural stem cells at the lesion site is considered, I would say, an appealing potential treatment because the hypothesis is that the transplantation may produce a replacement, of course, of the lost neurons, but also the creation of a more permissive environment, allowing maybe the axonal regrowth by host neurons, but also the progression of growth factors that in a certain way can converge in the creation of a relay station to transmit the information upstream or downstream of the lesion site. We were talking about stem cells for a therapeutic approach, but there is not a single kind of stem cell. And now I will focus the attention on this early neural tube formation. So initially the neural tube is formed by a pseudostratified layer of neuroepithelial cells. And these neuroepithelial cells are considered basically the founders of all the neuronal population present in our central nervous system. They are quite proliferative, symmetrically dividing, and also endowed with high plasticity. So these are the objects that we explored in our study to try to address the spinal cord injury treatment. Recently, we obtained a collection of human neuroepithelial stem cells derived from post-mortem embryonic human specimens brought from the neocortex and from the spinal cord. We also characterized with single-cell transcriptomics the profile of these cells and compared the nest cells cultured in vitro with the in vivo counterpart through isogenic matching of the donor and tissue derived. These cells also, interestingly, form very arranged structure that are called neural rosettes. So basically, the progenitors are arranged radially around the central lumen that correspond basically to the central canal of the developing spinal cord. They are quite homogeneous, they are highly neurogenic, and also karyotypically stable. Spinal cord neuroepithelial stem cells express a typical marker of caudal neural progenitors. In particular, they maintain the code of the transcription factor that are typical of the region, of anatomical region of the derivation. For example, they express, as you can see, OXB4 and OXB9 that are typical of this region, but do not express other markers of other central nervous system areas. Neocortical neuroepithelial stem cells share the same general neural stem cell markers, but they do not express the posterior marker as the spinal cord cells that I mentioned, but basically they express marker of the forebrain, in particular FOXV1, for example, and OTX2. So the first thing that I would like then to keep in consideration for discussion in a few moments is that these cells maintain in some way the memory of the region where derived from, also in culture, for a number of passages. Also, single-cell transcriptomics performed on nest cells confirmed that these cells are quite similar to the donor tissue. So you can see here the expression of a panel of diagnostic markers typical of early neurostem cells, radial glials, but also committed progenitors. And if you compare the level of expression of these genes on cultured nest cells, you can see that the profile is quite conserved, in particular for the early neural stem cell properties and also for committed progenitors. We also optimize a protocol for neuronal differentiation in vitro that consists in a quite long protocol, up to 3 months, that allow the differentiation of functional neurons after the withdrawal of mitogens and the supplementation of growth factor like BDNF and other molecules. And at the end of this process, we can see that nest cells are able to produce very, very important neuronal networks with very immature neurons. Also, a fraction of these neurons express typical markers of cholinergic motor neurons, as shown here by chat expression. But these cells are also tripotent because they are able to differentiate also in astrocytes, as demonstrated by the expression of GFAP, or even mature oligodendrocyte, as shown by the staining for O4. So we turn to our initial question, so how to address spinal cord lesions. And we believe that these cells had the right ID to be a challenge in a spinal cord paradigm lesion. So we basically derived the cells, we labeled with GFP, and then we transplanted into an immunodeficient mouse model that is a non-skid, that received also a dorsal hemisection lesion at the level of the thoracic spinal cord portion. After 10 days, the cells were transplanted, and we then analyzed the integration ability of the cells, the graft-host interaction, and also some functional tests. Here you can see basically that the spinal cord received a lesion, so a dorsal hemisection, as I already mentioned, and in green you can see the beautiful integration of these human cells in the spinal cord. So in green you can see also that a number of processes is extending toward the rostral part of the spinal cord and toward the caudal part, while in red you can see the expression of endogenous astrocytes that usually are also going around the lesion site, forming what is called the glial scar. If we have a look at the magnification, so it is the dorsal part of the lesioned spinal cord. So you can see here that cells basically are trying to fill the gap between the upstream and downstream part of the resected and hemisected spinal cord, trying to basically behave like a glue. And interestingly, from the grafted cells, a number of axons started also to extend in particular in the white matter, and also we quantified the number of processes, and more than 2/3 of the axons from the transplanted cells were in the white matter, only 30% in the gray matter, as a good distribution as endogenous fibers into the different compartments of the spinal cord. This is also demonstrating that myelin, so adult myelin, is not inhibiting the axonal growth, as usually this substance is doing with endogenous neurons, creating a very impervious environment for axonal regrowth. We also had a look at the extension of the processes starting from the transplanted cells, and we have a look both rostrally and caudally to the lesion site. For example, as you can see here, the extension of the axonal tract from the transplanted cells is reaching the medulla oblongata, but also down up to the conus medullaris near the coda equina. So these cells, after 2 months, are able to reach both sides of the spinal cord, the mouse spinal cord, after the transplantation, demonstrating to be able to grow along the 4-centimeter length of the mouse spinal cord. We then moved to see if the histological integration was also mirrored by a kind of beneficial effect in terms of recovery of motor behavior. We did some tests. So I'm showing here the open field hind limb locomotion test that basically is scoring the ability to move of the mouse through the BMS, so Bowes muscle score, that is basically showing that control mice that were lesioned but didn't receive any cell transplanted experience a very, very low spontaneous recovery. On the other hand, transplanted spinal cord nest cells are able to rescue for a significant level the motor behavior of this cord. In particular, the amelioration was not due to the different level of damage of the injury produced in the spinal cord, because we analyzed also the spare tissue in both in control and transplanted animals, and they were very comparable. We did also another test that is called the grid walking test. So let me try to show the movie. Sorry, one issue here. sorry. Okay. There is one issue. Sorry, let me try again. Okay, yes. Okay, so here is basically the quantification of the incorrectly placed steps on a grid. So you can see that lesioned mice have a severe effect on the very high limbs, but this also incorrect behavior is in some way recovered in transplanted mice. Oh, so let me put back the laser. Okay. We then asked whether the recovery, so the good integration and the recovery, motor recovery produced by spinal cord nest cells was dependent on their specific anatomical origin, or even the other population of neocortical cells that I already described were able to produce similar effect. So we also produced the same experiment and transplanted the neocortical derived neuroepithelial stem cells. But as you can see, these cells transplanted in the spinal cord were not able to spread, to extend the axons, and remain poorly differentiated. As you can see also the quantification, so very few axons were able to cross the border of the transplanted graft. And more remarkably, the cells were not able to produce any amelioration in terms of motor behavior. So this is important to reinforce the concept that not all the stem cells are the same. In particular, so the anatomical regional derivation should fit the target area of a transplantation to produce any integration or significant beneficial effect after transplantation. Also, to dissect the diversity of the spinal cord versus neocortical graft into the spinal cord lesions, we performed a global transcriptome analysis and compared the transcriptome from the graft extracted after 2 months from the transplantation, but also we include in this unbiased analysis also the transcriptome of proliferating spinal cord or neocortical nest cells, and also differentiated in vitro differentiated neurons with the same age of the graft of 2 months. We then calculated the general transcriptome profile, and in particular we focus on differential expressed genes. And in particular, among the top 50 upregulated genes, we found a number of markers that are correlated with good maturation. In particular, so maturin was one of the most abundant genes expressed in spinal cord graft, and also pleiotrophin, that is involved in axon elongation, but also other synaptic genes were quite upregulated. All these genes were, of course, downregulated or not expressed in the neocortical graft. What I would like to reinforce here is that also the regional markers, like for example OXB4 and OXB9, were maintained long-term, even in vivo after transplantation in the neocortical cells, or in the neocortical graft. Sorry, in the spinal cord, I'm confused. So in the spinal cord graft, we have still the expression of the OX genes, while in the neocortical parts, we maintain the expression of OTX and FOXP1, probably also explaining the diversity and differential activity in terms of integration and beneficial effect. We also validated through immunohistochemistry the expression of some of the data coming from the transcription analysis, and as you can see, we analyzed the protein presence of maturin and the pleiotrophin in the spinal cord graft. As you can see, there is a good expression of these markers that is lacking in the neocortical, basically, graft cells at the same time. So we can consider also this maturin and the pleiotrophin as the candidate biomarkers of good integration and maturation in vivo of the transplanted cells. So I hope that I convinced you that neuroepithelial stem cells derived from the spinal cord are a good tool to explore possibility of transplantation in the spinal cord lesions. Now I would like to move in the few next slides to the combination of these technology, so this approach, cell transplantation, where we can see that there is still room to improvement. So the combination with another approach that is coming from the nanomedicine, in particular, thanks to a collaboration with Vittoria Raffa Group, that is a leading scientist in the field of nanomedicine and stretch grow applied to neuronal regeneration. So basically, the idea is to decorate the neurons with the magnetic nanoparticles and then expose them to an external magnetic field, and basically the force is very, very low force anyway, is able to stretch and improve the axonal growth, but also the functional maturation. So the global design is to decorate both the injured neurons into the spinal cord, so the endogenous neurons, and also the transplanted cells with the magnetic nanoparticles, and then expose it to an external magnetic field. And so this combinatorial activity would reinforce the endogenous regeneration by spared axons, but also allow ordered, we can say, axonal growth and extension and improvement of the maturation and acceleration of the functional activity of the transplanted neurons. I would like to show here some preliminary data that we got recently by applying this stretch grow tool on spinal cord nest-derived neurons in vitro. So in short-term paradigm, nest cells exposed to for 48 hours are able to extend the axons, and there is a significant increase in the length of the axons that basically double in 2 days, while there is no change in the caliber of the axons, meaning that what we are assisting is not just a viscoelastic deformation of the axon, but is a positive growth by mass addiction. We also perform the same experiment in the long-term context by applying the protocol for 60 days in vitro. And as you can see, the stretched neurons are also increasing the connectivity and the total neurite area, but also there is an anticipation of the maturation as demonstrated by the expression of the mature marker RBFOX3. So the combinatorial activity of stretch grow is able basically to impress also and to propel basically the functional maturations of these neurons. I have no time for the moment to show also the electrophysiological properties, but the bottom line is that then after this good preliminary result obtained on in vitro differentiated neurons, we also moved to an ex vivo paradigm by transplant spinal cord nest cells into the organotypic slice derived from the spinal cord of P3 mice. Basically, we obtained this ex vivo paradigm, transplanted the cells on top, and then exposed the cells to nanoparticles and the external magnetic field for a certain temporal window, as shown here. And also in this context, we were able to assist to a process elongation dictated by the external force that is able to improve the elongation and maturation of the transplanted cells, even in an environment that is not just a reductionist approach in 2D culture, but even in an environment that is mimicking what is the real tissue. Just to summarize, so human spinal cord and cells grafted in the injured spinal cord differentiate into neurons and they integrate. They also produce an amelioration in terms of motor behavior through the formation of a novel circuit. And also, we define the idea of a good population of nest cells because neocortical nest cells are not able to produce the same effect. Transcriptional profiling identifies also potential candidate biomarker of good grafting and also optimal maturation. And stretch grow also is a new tool of nanomedicine that is able to enhance the axonal growth and functional maturation of the neurons. So we believe that the cell therapy and combined with the stretch grow tool are promising combinatorial approach to treat and address spinal cord injury. Let me thank the people of the lab that mainly work on this project. So Matteo, Francesca, and Alessio, and also the collaboration with the Vittoria Raffa groups and all the other people and the granting agency. Okay, so thanks a lot for your attention.
Thank you very much, Marco, for this novel approach in tackling the spinal cord injuries. Now I give the floor for the moderation of the panel discussion to Professor Alessandro Vercelli, and who is the scientific director of the Neuroscience Institute Cavalieri Ottolenghi of the University of Turin, and also the president of the Italian Neuroscience Society. And I also leave him the duty to wrap up the webinar. Please, Alessandro.
Okay, thank you, and thank you for to everybody for having made possible this webinar, which I found very interesting. And so I first ask if anybody has any question. I don't see any question in the chat. Let me see. No, at the moment there are no questions. And well, I would like just to start with a small question to Professor Tadahashi. Please, I just wanted to know, he showed us the results with the electrical stimulation. Did you try also transcranial magnetic stimulation?
Yeah, maybe. So first we start, since we are implanting the electrode, so we, as a first step to make it more efficient, we directly stimulate it in the cortical surface. But I hope we can transfer, as a next step, we'll move on to the less invasive transcranial magnetic stimulation. We first implanted the electrode for the recording, but at the same time, we use that electrode for the stimulation and found this. But yeah, as a next step, we are planning.
Okay.
This basic technique.
Yeah, thank you. Because there are a lot of things going on with the transcranial stimulation, and of course it will be much more feasible and acceptable for patient treatment, of course.
Yeah.
Okay. Then any other question?
Actually, I have one question. I tried to put the question, but I cannot upload it. So I have a question to Dr. Buffo. So actually how the neuron in the graft differentiate was really amazing. And in fact, but I do not know how much of the mechanism of the cell death in the Huntington disease. But do you expect these neurons can survive in the striatum of the Huntington disease patients? I don't know about the environment of striatum in patients. Do you think they survive?
This is a very important question and a concern that we are directly addressing now by grafting the cells also into a diseased environment. So we are employing mutants, genetic mutants, mouse genetic mutants to simulate the environment. It seems that the cells can survive at least up to 2 months. We don't have longer time points for the moment, and actually these experiments are performed by Linda Scaramuzza in Elena Cattanio's lab. So, of course, this is a real concern, and also we don't know how much neurons overexpressing or expressing mutated huntingtin will be capable, I mean, host neurons will be capable to wire the graft. So the model that we are using is, let's say, is very good to explore the best potential of an adult brain, which is with a specific loss in the striatum to integrate the grafted cells and to connect to the grafted cells. But of course, the disease per se is much more complex, and as you suggested and pointed out correctly, additional detrimental factor has to be taken in account. So ideally, perhaps a cell therapy should be combined with a genetic approach which is lowering overall the level of mutated proteins, so that also, I mean, neurons that are not primarily affected by the disease can actually go back to normal functioning and express their capability to connect.
Yeah, thank you very much. Yes, such combination would be very, very important. Thank you very much.
Okay, thank you. And I don't see any other questions, so I would continue to do some questions, to ask some questions. I would like to ask to Professor Kageyama about neurogenesis in the adult, because of course he showed very, very nice data on the rodents, but there are many concerns about the occurrence of adult neurogenesis in primates, and especially in humans. And I know we have a colleague in our institute who believes that in humans there are mostly progenitors, something like resting progenitors, which have to differentiate in the aged people. So what do you think about neurogenesis in the adult human brain?
That's a very important question. Yeah, we have not worked any human samples, so we don't know, to be honest. But we recently worked on monkeys, and the monkeys definitely neurogenesis occurs in the adult. So I think humans, I expect neurogenesis occurs human also, but it's still very controversial. Some data suggests neurogenesis occurs very actively and Alzheimer's disease is decline, but other data suggest that neurogenesis does not occur well. But as long as neural stem cells are present, then our method probably can activate these dormant neural stem cells very efficiently. So we hope neural stem cells are really present in the adult brain also, in human also.
And also, I wanted to ask you another question, because I read just the summary of a paper recently about the usage of cerebrospinal fluid from young people and injected into aged people. Do you think that this could help this neurogenesis in the brain or not?
Yeah, I think, yeah, those research definitely shows young blood contains some very effective agent to activate neurogenesis in the aged brain. So that's, yeah, I agree, that's very promising, but we don't know whether any specific factors are really important. So in our hand, well, such factors show rather weak activity compared to iPad. So iPad definitely, start neurogenesis like a very young brain. So maybe a combination could work better, or we don't know. But yeah, that's a definitely important issue in the future. Yes. Yeah.
Thank you very
Thank you
much.
very much.
Yeah, very interesting. Any question from the audience?
So if I may just raise, Alessandro, my hand.
Yeah.
Also, I see Annalisa with the hand, so probably she has the
Go
priority.
ahead, go ahead.
No, please go on. No.
Okay, so yes, I have a curiosity to ask to Dr. Kageyama, and which comes from my background. So, have you ever tried, or what happens if you overexpress the iPad in astrocytes or glial cells? So the question is this enough to elicit a neurogenic transition or to start some level of proliferative activity in quiescent glia? And what happens instead if such overexpression is induced in proliferative glia, which in some cases has been suggested, to acquire at least the potencies of a neural stem cell?
Yeah, thank you for a very interesting question. Yeah, actually, we infect iPad virus to astrocytes, but they don't change at all. So iPad cannot activate proliferation of resting astrocytes. So maybe, some astrocytes, so after astrocytes are activated, and if notch signaling is inhibited, they may change to neurogenic. They may have acquire neurogenic potential. So if astrocytes change to that state, I think iPad probably promotes proliferation and further neurogenic potential. But just resting astrocytes do not respond to iPad. So,
okay. So in other terms, if I understand correctly, so iPad acts on cells that have already a neurogenic potency and is like increasing the energetic level so to achieve full activation.
Yes, correct. Yeah. Yeah. So far, we only see the effect on stem cells. So dormant stem cells, iPad definitely shows a very strong activity, but otherwise, no activity. So yes, yeah, you're correct. Yes.
Thank you.
Thank you.
Okay, Marco, do you?
Yes, so thanks. I was also curious about your opinion, Professor Kageyama, about the adult neurogenesis, because there are debates about the presence in a human adult brain, in particular both in the subventricular zone but also in the hippocampus, that apparently drop around adolescent or even before in infants. So the thing is that there is debate, but the problem, to the best of my knowledge, is about the markers that are used to label progenitors. And there is one last paper that basically performed a single nucleus transcriptomics, analyzing both human, chimpanzee, macaque, and the pig brains. And basically, there was no a single, probably only one progenitor that was a hybrid, no? And so in human, basically, no, in adult, there is no evidence. But the interesting thing is that the marker that usually people adopt to label precursors in adult brain is double cortin. But the problem is that double cortin is a marker of early precursors of neuroblasts, usually. But in humans, apparently, there is an expression also in inhibitory neurons. So adult inhibitory neurons express double cortin. So this is not the right way to label progenitors. So maybe this is also the source of a debate. Okay. But so this is just to add something to the discussion. So the other things that I would like to ask you is about what is exactly the mechanism of this inhibition with iPad, and if this can be applied, then maybe it's conserved between species. Okay, so for example, to human. So you have any effect on the target and the mechanism? Thank you so much.
Yeah. So yeah, thank you for very important questions. About human, as I said, we have not examined human samples by ourselves. So I only imagine from the literatures. Well, our experience is mouse aged dormant neural stem cells. It's very difficult to extract RNAs. And even though we try to choose the same number of neural stem cells from aged and young adults, still the volume of aged neural stem cells are very small, much smaller than young neural stem cells. So I'm not sure whether the single cell RNA sequencing data really captures dormant small neural stem cells. So I'm not sure about that. So yeah, it's very difficult to get enough RNAs from these dormant aged neural stem cells. So maybe it's possible they might lose the RNA samples from those special types of cells. So that's one possibility. But so we are not sure about. So as long in our hand, we looked at monkey brains and we see Ki67 positive or BRD incorporating cells definitely present in a dental gyrus, and also found the D6 positive neurons nearby. So I think probably neurogenesis occurs in the monkey hippocampal dental gyrus. So so from that data, I expect neural stem cells are probably present in the adult brain, but it's difficult to detect, or maybe small number or small volume. But as long as they are there, then iPad treatment can activate those neural stem cells efficiently and produce many new neurons. So yeah, that's what I expect right now. And so we want to extend our method to monkey or eventually human patients in the future. Yes. Thank you for your important question.
Thank you.
Okay, so I think that there is a very last question from the audience to Marco, and it is to Marco, but it may be extended also to Annalisa, I think, about the tumorigenic potential of stem cells. Well, it's a long debated question, and can you comment on this?
Oh, okay, sure. I don't see the question. Maybe.
Yeah, well, it's.
I'm not familiar, sorry, with the WebEx platform. Anyway, so concerning the transplantation of new epithelial stem cells, so there are several reports that demonstrated that they are not tumorigenic, and they do not produce what is called also neural overgrowth. Okay, because usually with time they decrease, they integrate, and also we quantified the number of Ki67 cells after transplantation up to 2 months, and it is a strong decrease of proliferating markers, nesting, also had increase of neuronal and also glial markers. So there is no evidence up to now of the tumorigenic effects of these cells. Concerning other cell types, for example, more proliferative cells like embryonic stem cells or iPS-derived cells, the early progenitors. So the problem is that if there is no 100% differentiation toward the neural lineage, so pluripotent cells can remain in the tissue after the transplantation, and they can be teratogenic. So they can produce, this is the definition of pluripotent stem cells, so they can produce teratoma, to be honest. For this reason, so one of the most important efforts is to push homogeneously the cells toward early commitment, so toward the neural lineage. And so there are several ways. So you can just kill the cells that are still pluripotent, select with the FACS sorting strategies to remove any contamination, possible contamination. Put also suicide gene cassette inside. So if in case only one single cell can be undifferentiated and transplanted, with some strategies, genetic strategies, you can kill, so avoid any presence of pluripotent or highly proliferating cells. So of course, this is something we should consider, but the impact usually is low, okay? So to be honest, a lot of reports are saying that when you transplant cells of your beautiful neurons integrated, and the risk of neural overgrowth is usually very low. But of course, before moving to the clinics or even in the clinical trials, the purification should be 100%, okay, of the cells, of the right population of cells that we wanted to transplant, okay, not just mixture of heterogeneous cultures. But maybe now Annalisa can add something about that.
I would just like to add what I know from the Parkinson fields, where they have reached a stage to go to clinical trials, basically. And I know a bit about what the regulatory organism and institution ask to monitor the possibility that the therapeutic cells contain proliferative components or, let's say, multipotent component, if not pluripotent. And I can just comment that the tests that have been, let's say, planned and required are very strict, and they are multiple, meaning that there is a high attention on, let's say, the qualification of the cell preparation, which is ready or it is qualified to go to the patients, and all issues with respect to maintenance of any kind of pluripotency and proliferative activity, and even the nature of the progenitors that are planned to be grafted per se, has to be carefully addressed by means of several assays. And some of these assays did not exist before in the scientific community. So colleagues working on this, they had to invent how to prove what the regulatory authorities were asking them to be proved. So I think that the community overall is doing really its best to grant a high degree of safety for cells that are really going to clinical trials.
Okay, thank you, Annalisa, and thank you, everybody. I think that we already exceeded the time of the webinar, so I leave the floor to Professor Traversa in order to close the meeting. And thank you, everybody, for participating. I hope that, as I told before, this is only a beginning of our exchange of ideas, and we have many opportunities in the future to exchange ideas and maybe collaborations. Professor Traversa.
Yeah, I was saying that you could wrap up the webinar, but that's okay. That's I thank you everybody for thank you very much the speakers. Thank you very much Professor Vercelli and Nico for having helped in organizing this webinar. And I really hope that this will be fruitful for future collaborations between Italy and Japan. And I hope to see you, everybody in Japan. Thank you very much.
Bye-bye.
Thank you, everybody. Bye.
Bye-bye.
Thank you.
Thank you very much.
Bye-bye, everybody.
Bye-bye.
Bye-bye. Bye.
Bye-bye.